Hi everyone. I am pleased to have the opportunity to introduce one of our recent work published in iMeta. The title is Fungi Stabilize Multi-Kingdom Community in a High-Elevation Timberline Ecosystem. It is well known that forests constitute the largest carbon sink among all the terrestrial ecosystems. However, climate change and human activities pose threats to forest ecosystems through wildfire, drought, deforestation, as well as disease and insect pest outbreaks. Soil is the living skin of the Earth, and at least a quarter of the Earth's life is contained in it. A comprehensive investigation of the characteristics and structures of the hyper-diverse belowground biota is essential. Considering belowground biodiversity actively shapes above-ground biodiversity, drives biogeochemical processes, and give responses and feedbacks to global change. Timberline is a broad ecotone between closed forests and alpine grasslands or tundra, which is very sensitive to climate change and human activity. In the book of Alpine Treelines, the author addresses the role of above-ground biota in the formation and evolution of timberline. However, the role of hyperdiverse belowground biota is often overlooked. Here, we collected the neighboring soils of 30 mature Betula ermanii individuals along the elevations of Erman's birch forest on Changbai Mountain, which is one of the most well-protected alpine forests in Northeast Asia. Meanwhile, plant community composition and cover were recorded in the field. By plot survey, enrichment, culture, and sequencing, we obtained the community information of plants, bacteria, archaea, fungi, and nematodes. Using KIME2 and SPIEC-EASI methods, we constructed the multiple kingdom networks and single kingdom networks separately. Later, we did the downstream analysis by our software. Here, we hypothesized. 1. Multi-kingdom network construction would largely increase the complexity and negative edge proportion when compared with that in single-kingdom networks. 2. Species belonging to the fungal kingdom are crucial for the stability of multi-kingdom community association networks. 3. Fungal community would be the bridge of the multi-kingdom community, linking above-ground and below-ground communities. Firstly, we found bacteria and fungi are the largest groups in the multi-kingdom network, with highest node number, edge number, and mean connectivity, but lowest modularity. Compared to those in the single kingdom networks, the edge number and mean connectivity were largely enhanced. In particular, the increases in mean connectivity and negative edge proportion were the most significant for the fungal subset. At the node level, the normalized degree and betweenness of fungi were significantly larger than those of bacteria. It indicates that fungi owns more links to other species and more likely to play as brokers. More amazingly, the addition of fungi enhanced the network stability and connectedness of plants, nematodes, and archaea substantially. In addition, fungal links accounted for more than a half of total cross-kingdom links. Compared to the randomized links of fungi, fungi were more prone to linking with plants and archaea. At the community level, both MRM and SEM models show that the plant community strongly affected the fungal community, whereas the fungal community significantly affected the archaeal and nematode community, and vice versa. Clearly, fungal community is at the center of multi-kingdom communities, linking above and below-ground communities. In summary, in the alpine timberline ecosystem, we demonstrated that multi-kingdom network construction increased network complexity and negative edge proportion. In particular, fungal kingdom stabilized multi-kingdom network structure, as well as linked above and below-ground biota at community level. The findings greatly enhance our understanding of the potential roles and interactions of different soil biota and plants. Thank you for your attention.